Bit by bit, I'm going to show images and talk about central neurocytomas. Central neurocytomas are rare tumors, less than 1% of all intracranial neoplasms, and you should think of a central neurocytoma if you see a bubbly mass located intraventricular, especially in the frontal horns or body of the lateral ventricles, with a close relation to the septum pellucidum. Central neurocytomas are partly solid and partly cystic tumors, as you can see on these T2-weighted images. They present with hydrocephalus in young adults, and after contrast, central neurocytomas enhance, differentiating them from subependymomas, another intraventricular tumor. Central neurocytomas have calcifications. You can see that on these CT images. And macroscopically, they resemble oligodendrogliomas. And previously, central neurocytomas were called oligodendrogliomas of the ventricle or intraventricular oligodendrogliomas. However, central neurocytomas are not made up of glial cells and they don't have a 1p19q codeletion. Central neurocytomas are made up of uniform cells with a neuronal differentiation, hence the name neurocytoma. The calcifications that you saw on CT can also be seen on histology, and central neurocytomas are moderately vascularized, and if the vascular pattern is a honeycomb pattern, they resemble macroscopically and on imaging an oligodendroglioma, and sometimes it's more a perivascular pseudorosette tumor pattern, and then they might resemble an ependymoma. So central neurocytomas are thought to arise from neuroglial progenitor cells, and last year there was an article in one of these journals, part of the Nature group, where they showed the similarities between the cells of a central neurocytoma and the radial glial cells, and we talked about the radial glial cells in the first videos on this channel about the malformations of the neocortex. So you might remember that radial glial cells were these neural stem cells located in the ventricular zone. They give rise to the neurons and glial cells, and the neurons migrate along the radial glial cell towards the cortex. So that's about everything there is to say about a central neurocytoma from a clinical point of view. So the next minutes I will spend on talking about the septum pellucidum, because central neurocytomas often arise from the septum pellucidum. And there's this general incorrect perception that the septum pellucidum is an atrophic vestigial structure in humans that doesn't contain anything except some glial cells and maybe a little bit of white matter. But the septum pellucidum is an important structure also in humans. So if you think of the septum pellucidum, that is in humans stretched between the frontal horns of the ventricles, you can distinguish an inferior part which is called the true septum or septum verum with the nuclei in it, and there's the very thin transparent part that's called the septum pellucidum. And this thin septum pellucidum part is actually some stretching of the inferior septum verum, the true septum, that arose in embryology and in evolution when the frontal horns and the frontal lobe was expanding. And think of this cavum septum pellucidum that you might see as a normal variant. So that's the location of the septum pellucidum. And this is an overview of the embryology, and I'm going to cut it down for you because there are some important things in it. So in the 5-week human embryo, so that's really early, so you just have the 2 brain vesicles, and there's the preplate, so you have radial glial cells and neural progenitors lining the ventricle, and 1 layer of neurons on the outside of that. And in this 5-week-old embryo, if you make a coronal section just anterior to the foramen of Monroe, as you can see in this article from 2021 from Dr. Harvey Sarnat, you can distinguish several parts of this ventricular zone with the germinal matrix and all these neural stem cells. So to start with the easy part, in the ventrolateral quadrant, there's the ganglionic eminence overlying the striate, which is going to give rise to the GABAergic neurons. Then there's the ventromedial quadrant, and that's where the septal nuclei are. And the septal nuclei are continuous with the hippocampus, or the hippocampal primordium, and the hippocampus is continuous with the piriform cortex and the neocortex. So this is a 7-week-old embryo, so the ventromedial quadrant is the septal area, and in the ventrolateral quadrant is the ganglionic eminence, and these are histological specimens, so you can see the septal nuclei here. And I was wondering, but I couldn't find it, that if you would go more dorsal, whether the area of the septal nuclei is continuous with the cortical hem. So I don't have the answer to that question. But you can see the septal nuclei here, continuous with the hippocampus. And then this septal area is going to grow with the expansion of the frontal lobe, and the cells, all the progenitor cells and the neurons remain in the nuclei in the true septum, the septum verum. And then you have this stretched cell sparse part of the septum pellucidum with some sparse neuroglial progenitors. So these neuroglial progenitors, they can give rise to the central neurocytoma. And it is incorrect that the septum pellucidum doesn't have a function in humans anymore. If you look at the size of the septum pellucidum in humans, here superimposed on the size of the septum pellucidum in this prosimian, this half ape, with the dotted line, you can see that through evolution, the septum pellucidum has grown. So these open bullets are the different species, and the percentage of the septum of the total brain, and the closed bullets are the percentage of the septum of the telencephalon. And you can see that it remains the same throughout evolution proportionally. And if you compare the size and the complexity of the septum to the most basal insectivores, this classification is a little bit outdated, but you get the idea. Then you can see that in humans the septum is really advanced, and it's the most it has developed and progressed through evolution. So the septum certainly is not a vestigial structure. So the septum verum, the true septum, that's the bride with the wedding dress. The septum pellucidum is the train of the wedding dress with some sparse neuroglial progenitor cells and a lot of white matter tracts. And this is a neurosurgical journal indicating all these white matter tracts connecting different parts of the limbic system. We don't know exactly what they do, but that doesn't mean that they are not relevant if you don't know what the function of something is. So the septum pellucidum is a really interesting structure. So if the tumor arises from the septum pellucidum, the differential diagnosis is basically just the central neurocytoma. Whereas if it arises more from the frontal horns or the body of the lateral ventricles, you can also think of a subependymoma, and we will continue with that in the next video.